In a previous video, I talked about how we clone a gene based on using a cloning vector and inserting a plasmid into a bacteria and letting that divide and amplify our gene for us. But that can be a pretty labor-intensive and long process. So there's another one called the polymerase chain reaction, PCR for short, that was invented by Carey Mullis in 1983. And what it does is it allows DNA fragments to be amplified a billionfold within just a few hours. So it can be used for extremely small amounts of DNA, even a single molecule DNA, which is really nice, and it's less labor intensive than using a vector or cloning plasmid. The basis of it is basically PCR is replication catalyzed by DNA polymerase. Replication in this case has two essential requirements. So we're going to write the requirements down here. Two essential requirements are that the DNA template from which the new DNA strand can be copied and a pair of primers with a 3' OH group to the new molecule that can be added. So the first one is a DNA template. And the second one is a pair of primers with a 3' OH group. So if we look up here, we have this double-stranded nucleotide right here. So if we label it, let's pretend that this is the 5' end, 3', 5', 3'. How it works is you have these primers that are from 17 to 25 nucleotides long. So they're complementary to this known sequence in the DNA. So we'd have them, let's just say they're right here. So this is the 3' end. So you want to have the 3' end primer here. And then on this side, this would be the 3' end. So you have that right here. Now, how do you separate these strands? Well, we talked about this before. The DNA would be heated to about 90 to 100 degrees Celsius to separate these two strands. The DNA is then quickly cooled to allow, so actually I'll write this down here. Let's do the steps in orange. So step 1 is heat to 90 degrees Celsius or 100 degrees Celsius to denature and the double-stranded DNA. Then the DNA is quickly cooled. So step 2 was right here. Cooled to about 30 to 65 degrees Celsius. So the cooling allows the primers to anneal to their complementary sequences, but doesn't allow the DNA to reseal itself quite yet. So then the solution in step 3, we'll do in pink. Step 3, the solution is heated back to 72 degrees. That looks like a negative. Redo that. 70 degrees Celsius. And that allows the DNA polymerase to synthesize the new DNA strands that create two new double-stranded molecules. So if we have our primer right here, and our primer right here, and then we have a new strand coming in pink, like that. So that's synthesized. Now what you may be thinking is that DNA polymerase, there's no way that it can survive at a temperature of 90 to 100 degrees Celsius, because normal DNA polymerase in a human eukaryote, for example, denatures at 80 degrees Celsius. So what we use is called Taq polymerase, and Taq polymerase was huge for the discovery of this, and it comes from the bacterium Thermus aquaticus. So Taq polymerase comes from Thermus aquaticus. My apologies if I spelled this incorrectly. But they are found in the boiling springs of the Yellowstone National Park. So obviously they live in a hot environment, so they need to be able to replicate in a hot environment. So this is the perfect enzyme used for this type of experiment. So next step, the entire cycle would be repeated. So you'd have this single strand here with a primer. So you'd heat this back up. Step 1 again, right here, you'd have a single strand with your primer, and then this would get, let's draw it here again, and we'll do it on this side as well. So primer, And then you'd have your newly synthesized, I did this on the wrong side on this, but it doesn't really matter, you get the point. And then this can happen, so this has a logarithmic fashion of division, so each one of these is multiplying by 2 every time, so you can imagine that this cycle, if this takes, you heat it for about a minute or two, and then in step 2, you cool it for about a minute or less. You don't want it to be cooled too long or else the DNA will re-anneal itself. And then in step 3, you do it for a minute or less at 70 degrees Celsius. So this whole thing takes about 3 minutes, or 3 to 4 minutes. So if this is happening where you start with a 1 DNA strand to 2, by 20 cycles you'd have 1,000,000 molecules, and then by 30 cycles you'd have 1,000,000,000 molecules. So this would be amplified by 1 billionfold.